I am actually here in place of Rex Malmstrom, who runs our single-cell production pipeline at the Joint Genome Institute, and my supervisor, Tanya Wojcicki, who runs the single-cell program as a whole. And what I wanted to go over today was essentially, I'll give you a couple slides on the background of the Joint Genome Institute, but then talk about our single-cell production pipeline, where we use the echo, and then talk about a project that I'm just starting to work on now, where we're basically altering our single-cell pipeline, which the echo is sort of instrumental in order to try to extend the microbial tree of life. So this is a talk that is pretty different again, I guess, in that we're looking at single-cell environmental microbes for the most part. So the Joint Genome Institute, located in Walnut Creek, California, is essentially a user facility where we work really closely with our users. The mission is to serve as a genomic user facility in support of bioenergy. We would do a lot of work with poplar, switchgrass, bioenergy types of crops. Also carbon cycling. One large project is looking at wetlands in relation to carbon output in terms of carbon dioxide, methane emission, and then also biogeochemistry of interesting, quote-unquote extreme environments. That's more or less where I work. In terms of using some of these new technologies, single-cell genomics, metagenomics, to better understand how microbes essentially function in these environments. Here's just a quick slide on basically how the work is partitioned at the Joint Genome Institute. So again, we are predominantly user facilities. So the majority of work we do is interacting with the users, providing sequence data for them, but also assisting with the analysis part of their projects, depending on how much, what capabilities they have in their own labs. But then we also work with the Bioenergy research centers. We act as the facility to produce the sequence data for them. Some of these include JBay, the Joint Bioenergy Institutes, the Great Lakes Bioenergy Research Center, as well as Oak Ridge National Labs. And then we also have our Director of Science, which includes a fair number of things. Is where we do all of our benchmark studies in terms of testing new products from the wet lab side, even to testing our analysis pipelines. And then this is also the chunk of the pie, if you will, where we get to do some of our own science. And all of this sort of funnels into the different programs at JGI, so metagenome program, plants, fungi, microbes, and DNA synthesis science. So the single cell production pipeline falls within the microbe portion. So these are some of the typical environments that we work with, where we develop custom single cell and metagenomic projects, covering this diverse array of environment types. So, we work on bioremediation types of projects. So we did some of the sequencing for the Deep Horizon oil spill. As I mentioned, we also work really closely with folks who are interested in bioenergy types of organisms. So we do a lot of plant microbe interaction work. And then, all of these, really close to my heart, interesting environments where we try to understand the biogeochemistry going on there that's essentially governed by the microbes that are present, such as looking at microbial activity in subsea floor hydrothermal vents, in the volcano, Mount Ebris in Antarctica. We do quite a few studies in hot spring environments, certainly in Yellowstone, but also one of the studies that I'll talk about today is from a hot spring in British Columbia. And then, the recent interest in sub-glacial lakes, where certainly these are psychrophile type organisms. In addition, they're organisms that never see the light of day really. So I wanted to briefly introduce the concept of microbial dark matter, as this is sort of a focus of my supervisor, Tanya Wojcicki, where we're interested in quite a bit more than what we can culture on a plate. So as many of you probably have heard, with environmental microbial ecology, really, we can't really culture much from the environment. So, as you move out in the circles here, we have, a large chunk of uncultured phyla with genomic information, and then we have all these amplicon studies that have been performed over the last, 10 years, really, especially with the, Human Microbiome Project, for example. But then beyond there is all of the microbial groups that we've likely been missing due to limitations in these approaches. And for example, we know that there's a lot out there via amplicon studies, 16S specific approaches. However, the more and more we sequence with metagenomics, single-cell genomics, we're beginning to see new novel candidate phyla that seem to be missed by the 16S primers that are used in these types of amplicon studies. So that would make up the black region, the quote-unquote microbial dark matter, and then work in a collaborator of ours, Jill Banfield's group at Berkeley. She's really expanded the microbial tree of life using metagenomics and binning approaches. So that's extracting genome bins from metagenomic data sets. This is the candidate phyla radiation here. They are bacteria, but they're really, make up as much as 15% of the diversity of bacteria that we knew beforehand. And then there's also DPAN, an extension of the archaea that we didn't really know much about prior to these new technologies. So this brings me to taxonomic blind spots. So that is bacterial and archaeal clades that are either part of the rare biosphere. So that is essentially, the more you sequence, you still sequence the dominant populations in an environment. So it's really difficult to get at the rare biosphere. But more importantly to us, getting at the completely missed groups based on PCR-dependent studies. So here again is a three-domain phylogenetic tree. This is a genome tree. So, well, actually, yeah, this is based off of 16S sequences. And all of the red dots along the tree refer to phyla that do not have a cultured representative. So you can see there are many regions in the tree that don't have cultured representatives, and especially this region here is the candidate phyla radiation. So the extent of this is being argued heavily right now, but still, the main point here is that they make up a pretty large component of the bacterial side of the tree of life. So the technologies that have made this possible include amplicon sequencing, so that is 16S tag sequencing, shotgun metagenomics, and single-cell genomics. Both amplicon sequencing and single-cell genomics require the use of PCR primers, so they tend to be biased, given that these primers are supposedly universal, but no primer is, in fact, universal across all archaea and bacteria, for example. Yeah, so with the single-cell program, we're actually moving away from using 16S targeted single-cell genome production. What I mean by that is typically we'll have to amplify the 16S gene to determine what each of the cells are in a particular 384 well plate, give that to a user, then they come back, tell us which cells they want us to sequence. I'll get into a project where we're trying to do away with that step to basically decrease costs, but also it may help us get at some of these novel organisms that we're sort of hunting for right now. This is some work performed by a friend and colleague at the JGI, where she was essentially mining all of the metagenomes present in the databases currently, and then extracting 16S genes from those metagenomes, and then looking for primer mismatch sites from the dominant 5 primer sets. Like these include, this is what's used in the Earth Microbiome Project. I believe this one is in the Human Microbiome Project. And then, so the black bars represent 16S genes extracted from metagenomes, and the gray bars represent those from isolate genomes. And so what we can see is that when we're really interested in the diversity of environmental microbes, metagenomic data sets are now showing us that we would miss large proportions of the diversity that's present even using sort of these now traditional 16S amplicon approaches. So even with the best primer set, you might miss as much as 10% of the diversity in a given environment. The image over here shows the environments that represent the highest number of mismatches in terms of the 16S genes. So these are also environments where we might want to go sort of prospecting for novel microbial groups because they do have potentially weird 16S genes that would have been missed up to this point. So the main environment here is the deep subsurface. So we have collaborators, both terrestrial subsurface and the deep subsea floor, to perform sort of unbiased metagenomics, single-cell genomics to further take a look at these environments. So this is a outline of the single-cell production pipeline at the Joint Genome Institute. So as I mentioned, in a sense, this is a targeted approach where we use 16S screening as part of the pipeline. To begin with, we do the majority of our work using flow cell sorting, but we're also getting involved with microfluidics and Raman spectroscopy to look at functional genomics or functional single-cell genomics as well. And then the second part of the process is the amplification of, or whole genome amplification using MDA, because again, these are single cells aliquoted into a 384 well plate. So that is still, unfortunately, a necessity. And in the process, so we take advantage of the echo for preparation of the MDA products. And then from that point, we move on to our targeted 16S, still using Sanger sequencing for this, in a sense, because we don't have the need. The number of 16S sequences that we need isn't all that much, so we actually outsource that part of the workflow. Yeah, so then we typically will get 16S sequences back, say from a 384 well plate. Analyze that, determine what each of the organisms are from the 384 well plate, provide that to the user, and then they'll tell us which organisms from that plate that they want to get sequenced. And then there are a number of basically MDA products that are successful, but we get a lot of 16S, what we call 16S negative, or 16S during the 16S screening process, where the 16S screen doesn't actually work. But once we have the the 16S data back from or which microbes the users would like to screen, to get shotgun sequencing on. Then we move forward with shotgun sequencing. We run these through our assembly pipeline and annotation pipeline, and then get that back to the user. So now I wanted to talk about a project that I've been working on specifically, and this is to remove essentially the 16S part of the process in our pipeline. Again, to reduce costs, but also to look for microbes that we may not have seen before. And so instead of using the 16S gene to determine what each of these microbes are, we're taking sort of whole genome approaches by identifying conserved marker genes to come up with whole genome phylogenies. And then combining this with shotgun metagenomics. And again, this is to sort of expand the microbial tree of life and help us in terms of our cost-saving abilities. So this project that I wanted to just say a few words about is sort of a test case of this pipeline where we're working together with Peter Dunfield, a faculty member at the University of Calgary, and one of his main field sites is in British Columbia, where he has access to this slightly alkaline, high-temperature hot spring. And he's been able to find basically a bunch of candidate phyla within this hot spring already, and so we want to use this approach to see if we can come up with more potential branches on the tree of life. So to do this, and this is still very much so in progress, we have a batch of 16S negative, so that is cells that cells that pass the MDA amplification part of the process but failed 16S amplification for whatever reason. This could be due to, as Banfield and Berkeley has shown, the 16S gene from some of these novel microbes can have large insertion sequences where essentially the amplification of the 16S gene just won't work because the 16S gene is too large. And then there are also, as I mentioned, a number of primer mismatch sites potentially on the 16S gene. So we have around 300 16S negative single cells currently. We also have a bulk sample metagenome that we're comparing to. And in progress now, this was sort of my request, we want to compare these to essentially a proportional a number of 16S positive single cell genomes from the same sample to look at structure, function, and then really the phylogenetic novelty in this environment. So shown here is just essentially a quality control slide to show us that from this preliminary data, we essentially are getting good quality genomes. So this is just assembly statistics, genome quality. We're looking at marker gene-based completeness, assembly size, scaffold size. In gray are all of the 16S negative single cell genomes that we've sequenced thus far. We haven't sequenced all 200 of the 16S positive genomes. Hopefully when I get back I'll have those to play with. But we have 20 of them in yellow here. And really just the take home here is that there doesn't seem to be a reduction in quality in terms of assembly statistics with this 16S negative approach, which is great because now we can begin to use this new approach if collaborators want, just to sequence everything, get single cell genomes on everything in a particular environmental sample without the 16S screening part of the process. I also mentioned that colors didn't show up so well here. That we have a bulk metagenome that we're comparing to. We can use these as population genomes to compare our single cell genomes to. So this is just a quick slide showing that we have about 17, 18 genome bins that we can extract from the metagenomic sample. And some of them are fairly complete. And then these are just principal coordinates plots showing the basically the separation of the genome bins from one another. And then here, this is sort of the meat of the work that I'm doing at this point. And this tree will continue to evolve. But let's see, so we're looking at the composition and phylogeny of this particular hot spring with, around 300 16S negative single cells, the bulk metagenomic sample, as well as the 16S positive genomes that are en route, but we do have the 16S genes, so we can get an idea already of what the composition will be from the 16S positive data, as well as where they'll fit on a genome tree. So first, what we can see here is that the composition is quite variable between each of these three techniques. We seem to be missing entire groups with, say, the 16S approach or the 16S positive genomes, which we'll be getting back soon. So we're missing a lot of these Bacteroidetes, and yeah, that's the main group that we're missing. So if we look over at this full genome phylogeny, so this is kind of cool for me, actually. I'm just getting into this world in a sense. But basically, we took all genomes in NCBI, so around 60,000, reduced that based on extracting the 16S genes, clustering them, this tree using 85% sequence similarity clustering. So reduce that to about 500 16S genes that could then be mapped back to their genomes, and then we took those genomes, extracted all the proteins, and then from all of the proteins extracted the conserved markers that we want to use for a concatenated marker gene alignment and phylogenetic tree production. So what we can see on the tree are first that we have large numbers of, this is really sort of one of the most, in terms of number, one of the highest amounts of single cells that are sort of nearly identical. And I haven't done the analysis yet, but what we're planning on doing is doing sort of population level genomics on this group, which is the Aquavicae, once was a candidate phylum, as well as this group here, which is the Bacteroidetes. Sister phylum is the Ignavibacteria, or sister group is the Ignavibacteria, and then actually clusters together with the new candidate phylum within there, which has been given the name Kryptonia. So essentially, we can try to look for, which is a little bit new, looking for SNPs within single cell genomes. And then in relation to looking for novelty on the tree of life, we have these particular regions, which I have highlighted as subtrees on this slide. And really what we want to focus on here are any particular branch on the tree where the DC4 or Dewar Creek 16S negative single cell genomes are in blue, the metagenomic bins are in red, and then the 16S positive genomes would be in yellow. And so any region where we only have blue and red dots are interesting in that we would miss them with amplicon-based approaches. So we have an interesting group here in the chloroflexi, another interesting group here where we only have single cell representatives that we think are this candidate phylum S2R29, as well as another potential candidate phylum here in, related to the acidobacteria, but may be their own group, and then also within the Crenarchaeota. So where do we go next? Well, once I get these 16S positive genomes back, we can extract all of the 16S genes from these genomes say even from the 16S negative single cells, they obviously still have a 16S gene there, but get at the reason behind them being missed in the Sanger sequencing part of the pipeline. So to see then again if they have mismatches to the common primer sets that we're using in the Sanger sequencing step. And then also look at metabolic reconstructions, and then potentially do sort of the population level genomics on these two groups here that I have on this tree. And then lastly, I just wanted to bring up, give a plug for a couple of the programs at the Joint Genome Institute. So first there's the community science program where we do large-scale proposals that usually incorporate a variety of these pipelines, including metatranscriptomics, metagenomics, single-cell genomics, and then small-scale pipelines if you're just interested in a small set of samples using one of these tools. We also have an important visiting scientist program and pretty active postdoctoral fellowship program. And then finally, I've only been at the JGI for about a year and a half, but I've been to a couple of these meetings. They're pretty interesting because we get all of our users to come to these meetings for invited talks, but then we also have workshops on our technologies, but also our analysis pipelines, how we do our analysis pipelines working together with the NERSC, our High Performance Computing Center. So with that, I'd like to thank everyone involved in the single-cell production pipeline, as well as some of the project managers that I've worked hand in hand with. So thank you.